From the Oregon State University Extension Service, this is Pollination, a podcast that tells the stories of researchers, land managers, and concerned citizens making bold strides to improve the health of pollinators. I'm your host, Dr. Adoni Melithopoulos, Assistant Professor in Pollinator Health in the Department of Horticulture.
As we've heard in previous episodes, the bees that we find in urban landscapes are of intense interest not only to researchers, but to gardeners, landscapers, people who maintain parks. And the one thing that has become clear is that it's a very complicated system. There's a lot of things that are kind of underlying or driving the diversity, the patterns of diversity that we see in urban areas. And that's why I was so excited to finally welcome to the show Dr. Elsa Youngsteadt. She is an assistant professor of and extension urban ecology specialist at the University of North Carolina. I've been long admiring her program, which is really doing top-notch extension, but really great and nuanced research on bees in urban areas. So in this episode, we're going to hear about some of those factors that go into potentially driving diversity and some of the uncertainty we have around those drivers. And then the second part of the episode, we're going to tackle the most controversial of all topics, how to measure bee diversity, specifically something that's come under a lot of scrutiny recently, these bee bowls. How effective and biased are bee bowls at predicting native bee communities? So we're going to get into a lot of cans of worms in this episode this week with Dr. Elsa Youngsteadt on pollination. Welcome, Dr. Youngsteadt, to Pollination.
Thank you. Thanks for having me.
Now, we've had guests who have talked about the remarkable bee biodiversity in cities. And I wondered if this also applied to North Carolina. Are the bees in the cities the same ones you see in agricultural settings or in natural plant communities? Tell us a little bit about it.
Yeah, and I actually often wonder the same thing. Like, does this apply? Because when you ask yourself, like, what does it mean to have remarkable bee diversity, right? That suggests there's more than you expected. But then what were your expectations? And so I often see, studies of urban bees that conclude like, wow, this is more diversity than we thought would be here. But we never really know how much we expected to find. So I actually find that kind of a difficult question to answer. Just to put some numbers on it, like most of my work is in Raleigh, North Carolina. Some colleagues have also worked in Durham. So we're in this like Piedmont area of central North Carolina. That's overall a pretty disturbed, developed area. But between like Raleigh and Durham, we've got a total cumulative species list of maybe 130 or so in these kind of urban and peri-urban habitats. And that's out of, 5. We're also working on a state checklist for all of the species ever recorded in the state of North Carolina. That's up to about 560. So I don't know, is 130 out of 560 remarkable or not? It certainly seems like pretty good. Like we've got a good chunk of bee diversity in these urban areas, but there's definitely species that I never see in urban areas that I do see in some more specialized or pristine habitats, like nice, frequently burned sandhills habitat. There are some species that turn up there that I don't think I would ever see in a city. And certainly the composition changes, like the bees that you see, there are some urban winners, like specialists on violets. You will find far more of them here in the city than you do even in a beautiful forest full of spring ephemerals in the spring. So it's kind of a mixed bag. There seem to be winners and losers, but there's no doubt that cities are definitely supporting some solid chunk of bee diversity.
I love your nuanced answer because it is actually a really hard question to answer. And it's like, what subset of the whole fauna should we have? Like, it's unclear, before disturbance. And so you are left with this. And I love the example of the violets. I was thinking, so many people in urban areas plant sunflowers that, the sunflower specialist may be like, punching up above what you might find in a natural plant community. So like it is a very kind of nuanced question to ask. I always start with the nuanced ones.
Wait, what? Sorry,
I always start with like the trickiest. Oh no, it's fine. I start with the tricky question. So but let's get even trickier, because you've got, as you pointed out, in this urban and peri-urban area, you have a certain amount of diversity, and I suppose that area is not equal. So you would have like a park, there would be an industrial area. You have wealthy parts that where people have a lot of resources that can be brought to bear on gardens, and less affluent parts where the landscaping is minimal. So how do the patterns within the urban and peri-urban area influence the patterns of bees that we see?
Yeah, and you just hit on a couple of big, important, and different drivers. Like from the bee perspective, flowers are kind of the big obvious driver of bee abundance in cities. You go to like a botanical garden or places with intentional pollinator gardens, you're going to find more bees than if you go and try and sample on a monoculture turf lawn. And the way flowers are distributed in the city is driven, partly, like you mentioned, probably by socioeconomic factors, by wealth and the ability to sort of invest in ornamental landscaping. And I don't have sort of the bottom line on the wealth and bee diversity question yet. I know there are a lot of projects ongoing. I've heard informally from other students and other labs who have ongoing work in that area. My lab is one of those. So I'll mention a PhD student in my lab, Kate Gorman, who's got an ongoing project trying to kind of unpack whether this so-called luxury effect exists in bees, and if so, kind of what's driving it. Because you could actually imagine it going two different ways. Like I mentioned, flowers are one of the big drivers of bee populations, but then they don't just have to eat, they also have to nest somewhere. And nesting resources could also be limiting populations. We know the majority of our bees are nesting in the ground. Ground nesting resources are kind of slippery. It's hard to know exactly what any given species is looking for. You can't just go out and survey and be like, oh, yep, this is good ground nesting habitat, the way you can say like this is good flowers. And you can also imagine that, but we do think in general that like access to bare soil is a basic prerequisite for ground nesting bees for most species. And you can imagine that access to bare soil also could vary across socioeconomic gradients, across investment in landscaping. So you might have a lot of flowers in wealthy areas, but you might also not have a lot of bare soil if you're also seeing a lot of investment in irrigation and turf. So those resources could potentially get kind of decoupled. So Kate's doing this project across residential yards in Raleigh, across the range of sort of median income groups in our census blocks, to try and get at how those both aspects of, both kinds of bee resources may be shifting across socioeconomic gradients, and then what the actual upshot is for bee diversity. Maybe it peaks somewhere in the middle. So that's, great question. I hope we'll have some better answers soon.
That is a fascinating, the way that you frame the problem is fascinating to me. And I guess as Kate's work comes to fruition, you may see, I'm sure even, gradients of affluence can be really sharp and they can be subtle. And so you do have those effects. And I did wonder, I wanted to pick up, you mentioned in your answer that I thought it was a fascinating point that, we often focus on floral resources and we have ways of measuring that number of bees, diversity of bees visiting, but that there's this real problem being able to assess the quality of nesting resources. How is Kate trying to tackle that problem?
Yeah, she's been through a couple of iterations. Like the first field season, she went out and under my perhaps misguided influence, we just randomly placed emergence tents on different ground covers in these yards, on turf and on bare dirt and on mulch, because we really, we always hear the conventional wisdom that they've got to have bare ground, but is that really because that's the only place they're nesting, or because that's the only place you see them nesting and so you think that's where they are because they're just harder to detect? So we were going to try and use random placement of traps to get through those assumptions and see if they're right. But she caught, I think, like 2 bees all summer in those emergence tents, which maybe I should define what an emergence tent even is. They're starting to get used more often in bee research with mixed results similar to ours. But it's basically a little tent. Ours are, I think, 60 centimeters on a side that you pin down over the ground. And then there's a collecting head at the top. So any bee that is in a nest in the ground at the time you put the tent down, if it emerges either because it's a brand new bee starting its life cycle above the ground, or because it's a female who's coming in and out of that nest and was in the ground when you put the trap down, they'll fly up into the collecting head in the top of this trap and get documented. And it's a pretty common result for people who've tried to use these that you don't get very many bees, that it would be kind of a brute force effort to get a strong pattern using them. And we tried, and it seemed like the brute force was going to just be more than we could take. So what Kate's done the second summer is just visual searches for nests in all of these habitat types on transects in her yards. And then when she finds a candidate nest, any little hole in the ground, she goes and puts a trap over it to see if it was really a bee nest or something else, maybe a wasp or some ants or anything else that digs in the ground.
Oh, that's a great idea.
That has hugely increased her success rate at actually detecting bee nests. A lot of what she finds really are bee nests. And then, of course, we're still identifying them. We don't know yet if there's really, more nests in a particular part of our socioeconomic gradient or in particular parts of yards. But that has definitely stepped up our ability to document nests.
Well, we look forward to seeing the results. That is a great solution. And I have to admit, I have colleagues here at Oregon State University that have taken the same method of putting these tents up and really not getting a lot of data from them. So this is a really great solution of kind of like combining the two together.
Yeah, I still worry about the bias of like where you can actually see nests more easily. And another solution, and we can talk about this or not if you want, but another solution actually is one that Mike Ulishin and his colleagues in Georgia came up with, which was to just like rotate the location of the traps periodically. And they greatly increased their bee detections that way, rather than just put them on one spot and leave them for the summer. If they shuffled them, I don't remember, every week or so, they caught a lot more bees. And we were going to try that, but working in cities is weird. Like when you're working in somebody's yard, they don't necessarily have, you can't just say, okay, I'm going to come in here. I'm going to put this here this week, and then I'm going to put it here next week. Like there are restrictions. So we came up with our compromise.
Well, and I have to say, on that point, I know, there's a very nice mining bee nesting bed by the Hoyt Arboretum in Portland, and it's all on turf. And you can see them, they just pop out of the ground. And then if you come back later, it would just look like grass. So like it is a little bit, these biases are maybe unavoidable. It may be impossible to get anywhere without some bias, but nevertheless, keeping that in the back of our mind is like it seems like but not being paralyzed by it.
Yeah.
Okay. Well, that's a very exciting, and it's really exciting to see the nesting component come in, and kind of like making this picture much more complex. And just to raise the complexity, the other thing, when I think of cities is I think, there's variability. Some places are completely exposed. Maybe there's a new set of plantings for, some small trees, but it's sunny and hot and everything turns brown. And other places are these huge mature trees that cast a lot of shade. And I imagine with bees, shade is a kind of double-edged sword. It kind of cools and they may not want to go there, but at the same time, it can get pretty hot, especially these days.
Yeah. That's a great observation. And I agree with you that if you're going to go out and look for bees, like in the shade is not where you typically look. They're not usually, like, a lot of plants that produce nectar and pollen also are not shade loving. So I'm not sure how much of it is like where the bees are comfortable versus how much of it's just where their resources are. But the presence of big trees, or maybe I should start on the other end, the absence of big trees from like developed downtown areas is one of the major contributors to the urban heat island effect. Like that, if there are no trees, it's gonna be hotter. And at least this is coming from my biased perspective of like working in eastern forest ecosystems. If you're talking about, desert or grassland cities, things might be different, but in a forested ecosystem, the absence of trees in cities is one of the things that makes them hotter than their surroundings. And we know from other studies we've done in Raleigh that some bees can take it hotter than others. The ones that can't take it real hot, you don't find them in these urban heat islands. Their populations tend to decline in the hottest parts of the city. So even though you're not going to actively find bees foraging in shady areas anywhere, the fact is the shade is like cooling the air, and then that cooler air is still mixing with the air over your sunny pollinator garden. And overall, that habitat is not going to be as hot as it would be in a downtown where their trees are not making any cool air anywhere. There is no cool air to mix with, and it's just getting hot as blazes.
How does this cash out? Like what's the balancing point, and how important? I mean, I imagine one component of trees is the nectar and pollen they produce. That there's some great nectar.
That's the other one that I didn't even acknowledge is the trees themselves can be huge resources, especially in the spring. Like in our biome over here in the eastern U.S. anyway, a huge amount of the nectar and pollen resources are coming from trees themselves. And even in forests in the early spring, before the leaves fill in, the forest floor can be a great source of floral resources. And those tend to be also pretty, ephemeral bees who are just active for that little amount of time in the spring. And then you don't see them foraging in the shade once the trees fill in. But from an urban perspective, kind of what's the upshot? Like you got to have some trees to cool things off, but you can't have too much shade or the rest of the floral resources that the bees need for the rest of the year after the trees stop blooming, like aren't going to grow in the shade. And so there may be, it's hard to retrofit a downtown with big trees, as you already noted. Like if you put them in a miserably hot habitat, they don't tend to take off well. There can be some nurturing that can definitely increase canopy cover in certain urban areas. And that's one strategy for reducing urban heat islands, as long as you're, also keeping other floral resources around. There are also other urban cooling strategies, things like, light colored rooftops or other ways of reducing the heat island that can be effective. I think the other thing I want to comment on is that it doesn't just matter how hot it is. Like I said, some bees can take it hotter than others. But then that also is not the only thing that matters. Like you can think of it almost like a pesticide risk assessment, where you're interested in how sensitive a bee is to an insecticide, but also how much it is exposed to that insecticide. You can kind of think of heat similarly, because you might have a bee with a very low heat tolerance, but who's very good at thermoregulating, or whose body never gets very hot, or is only active at cool times of day to begin with.
Oh.
And that bee might not be at great risk of overheating, even though it appears to have a very low heat tolerance. Whereas some bees with a very high heat tolerance might still be actually cutting it pretty close. Like we see that with bumblebees. When we measure their heat tolerance in the lab, it's kind of medium. It's not the lowest of all the bees. But when we measure their body temperatures in the field, it's pretty high. They actually cut it pretty close. And so we might worry more about them than some of the species who actually have a lower heat tolerance, just because those other species aren't getting as hot in the first place, even in the same habitat.
And you've evaluated this. You've taken different species and kind of started to crack the nut of, how different bee taxa might respond to heat. Tell us a little bit about, you told us a little bit about bumblebees being surprising in that, they tolerate the heat, but they're not great heat dumpers per se. What are some of the...
actually have some pretty remarkable like thermoregulatory mechanisms. Like there are elaborate things they can do to, like, even though they've got this very hot flight engine in their thorax to shunt that heat off into their abdomen and avoid deadly temperatures, they still cut it kind of close. And interestingly, one thing that we found in my lab, a student in my lab, Malia Nomchik, who's moved on to do a master's now, but she was curious whether bumblebees carrying larger pollen loads actually get hotter, which sounds very intuitive. Like, we carry heavier things, we get sweatier. But when Malia wanted to start that project, I was like, oh, we're never going to detect that in the field. Like, that's a great question and you should try it, but don't hold your breath. She found a difference, like by going out and catching bumblebees with different pollen load sizes,
really,
and controlling for their body weight and the environmental temperature around them. They got about 0.07 degrees hotter per milligram of pollen, and they were carrying up to like 30-ish milligrams of pollen. So overall, like about 2 degrees Celsius of their body temperature could be due to like how much work they were doing just by carrying resources back to the nest.
This is fascinating because it isn't just, I love how how you think about this problem. It's not merely this thermal temperature that they can tolerate, but it may also be that they compensate by lower loads. And so the colony's reproductive rate is impacted, even though the bee doesn't like croak.
Exactly. I don't think for the most part, we're not going to see bees like actively dropping out of the sky because they let themselves get too hot. They've got to do something to avoid death. And that something is probably going to have impacts elsewhere in their life cycle. If it means bringing back less resource to the nest because you've got to carry a smaller load, or because you just have to stop activity during a certain time of day. It's gotta add up somewhere in the life cycle.
Fascinating. Well, let's take a quick break. I want to come back. We have another topic that I want to pick up with you in the second half that you've dangled in front of me, and I think it's going to be fascinating for the listener. So let's take a quick break and we'll be right back. Well, welcome back. So I just switching gears. I guess it's not totally switching gears because we trying to estimate bee communities in these different gradients, in urban areas where there's shade and, requires some kind of sampling method. You have to be able to catch the bees, and many times in a lot of cases, you have to be able to look at them under a microscope. So you need a specimen. But there is this question of, the different methods and the biases, really predicting what's out there. And I know there recently, one of the most common methods that people have been using for years, we've used it for years as well, are pan traps, these little colored traps with soapy water. Why don't you describe the pan trap and tell us, why you became interested in the biases associated with them?
Yeah, so pan traps, they come in a variety of sizes and colors, but the ones that we use and that I think are most common are probably 10 centimeters diameter or so, and they're little souffle cups that are painted with different colors, usually a UV reflective blue, yellow, or white. And the general idea is that bees are attracted to these because of the color, but then you've filled them with soapy water, so the bees that come to check them out fall in and drown. So it's a mean trick, but it's become a popular sampling method because it seems to take out the question of observer bias. Like if you send a bunch of people out into a habitat with nets, different people might come back with different samples and you would get a different picture of the underlying bee community. And the idea with pan traps is that maybe it takes out some of that observer bias and gives you a more consistent yardstick of what is there. But then there's been growing debate over the past few years that, are pan traps really so unbiased? Like maybe they don't work well when there's a lot of flowers. Maybe they overrepresent sweat bees. And bee biologists have noticed for a long time that, yes, if you go netting versus if you put out pan traps versus if you put out a vane trap, each method is going to give you a different picture of the bee community. But which one's actually right? Like we know each method is probably biased, but there hasn't been much done to try and actually quantify those biases or correct for them to get a clear view of what the underlying bee community really is. What is the relative abundance of the different species? How common is any given species? Just because it's the most common in your bowls doesn't mean that it's the most common, in the habitat.
So people had hunches, but they never quantify. Well, was it just unquantifiable? Is this something that's beyond the questions of science? It can't be validated?
Well, no, it's not. We tried to do a mark recapture study to get at some of this. In fact, we didn't just try, we actually did one. And so mark recapture is a method that's been common in wildlife biology for many decades, and it's even used for some insects, but hasn't been brought to bear a lot on bees. As part of our project, we did a lit review and found 20 different studies over the decades that have tried to do population estimates to estimate the abundance of bees in a habitat using mark recapture. So it's certainly not unheard of, but it seems to be a little bit neglected in the current debate of different biases, because this gives you a method to actually quantify the detectability of each bee species or field identifiable bee taxon in an environment. So you go out and you mark bees for a day, you let them go again, then you come back the next day, you see how many of that bee is marked versus unmarked, and it gives you an estimator then of what proportion of the population you're actually seeing on any given day. Let me get this straight.
So you mark the bee, you go out, you catch the bee, you say, this is, this is the best taxonomic unit I can do visually. You write it down and you put a mark on it. And then you put your pan traps out, and then you see, well, am I seeing the same proportion of marked bees in my trap? Am I seeing a lot of them in my trap, or am I not seeing any marked bees in my trap? Is that the idea?
Yeah, that would be one way of doing it. To actually get your mark recapture population estimate, you have to sample repeatedly non-lethally. So we would go out 3 days in a row to try and re-net bees that we had previously marked. And so each bee got kind of a day-specific color. So we would have a capture history for a series of individual bees. And that let us estimate the population for each of those taxa. So that was one view of the community composition that should have been bias corrected for detectability because we did the mark recapture. And then separately, the following week, once we had done those non-lethal estimates, then another team went out and put out the bee bowls and also did timed netting. And then we were able to look at the community composition in those samples and see which one best matched the bias corrected estimates from mark recapture.
Okay, so you've got the community structure, but you should get the same proportion of marked bees in the two different detection methods if they were reading the same thing. I'm not an ecologist, so this might be a little bit above my pay grade.
Yeah, and that we didn't get a lot of marked bees the following week. And I think we might need to fiddle with the assumptions in the model if we wait that long between capturing episodes, because we had two different teams in the field. There was the team doing the mark recapture, and then there was a separate team doing the pan trapping and netting. And there were different amounts of time in between sometimes just because of the weather.
Oh, sure.
So I think we would need to switch to an open population model that says that bees could die or immigrate during that intervening time, which over our 3 day, boom, 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 mark recapture episodes, we were able to say that we assumed that the population is pretty stable during that short time period, that bees are not getting added because they just declosed, or they're not disappearing because they departed or died.
Okay, but everybody listen.
There would be a way to do what you're saying. I don't even think about what that is exactly.
Everybody's wondering, though, how do you mark a bee?
Yes. We just use a little paint pen. So we put them in a bee squeezer, kind of like a queen marking cage.
Yeah.
To restrain them, and then just put a little paint dot on their back using a paint pen. And a lot of bees seem to not mind that process at all. We've marked honeybees and let them go, and leaf cutting bees, and they'll just go right back about their business, not leave the area, just continue foraging as if nothing had happened.
Okay, so what did you find?
So we found that the community captured in bowls was quite unlike the community we estimated using mark recapture. So indeed supporting the idea that, like, yes, bee bowls are quite biased. They're not really giving you a good picture of the underlying bee community that you would get if you used a bias corrected method like mark recapture. We were interested to find, though, that the community captured by hand netting correlated quite well with what we found using mark recapture. And that was especially interesting because different people were doing the netting on different days. It's not like the same person did mark recapture and then also went out and just did timed netting. These were different individuals. And so, of course, I'm not going to say that individual observer bias doesn't exist, but it was much less of a concern than we thought it was going to be in this particular study.
I imagine some of this must be great for people listening who are like, but I really, I understand the terms of this debate, but I'm worried about observer bias. But I imagine when you tell it, there are some of our best collectors, Dan and Michael, they're brothers, and they collect completely. They're real clear about this. Is like, Dan sits and waits at a flower and really targets the smallest bee he can find. And Michael is a kind of grazer. He just kind of like is going around. But I imagine if you gave Dan and Michael a very set protocol and said, this is what we want you to do, that would eliminate their kind of, their tastes for bee collection.
Yes, exactly.
Fascinating. Well, this is really great. I'm really looking forward to seeing this work as it develops. Let's take one more quick break, and we have a segment we do with all our guests. We want to know if you have a book recommendation, your go-to tool, and most importantly, your favorite pollinator. So we'll be right back. Okay, we are back. I'm very curious. What I can see your bookshelf back there. In fact, I'm curious if you have a book recommendation for our listeners.
I mean, the book that comes to mind is one I'm sure that many of your guests have recommended, but The Solitary Bees. And I should have looked up the correct order of authors before.
Oh yeah,
before I went to recommend the book. Danforth, Minkley, and help.
Jack Neff.
Neff. And is it that order? Danforth, Minkley, and Neff?
They're going to write us back. They're going to be like, you need to publish in erratum. Anyways, a great book, though.
Yes. I wrote a review for it once for American Entomologist, which was a great incentive to actually read it from cover to cover in a fairly continuous sitting, which I'd completely recommend to anybody else who might want to read it too. It's a great reference book. You can open it up and, find whatever species or topic you want to know about and read those details. But also it's a great read end to end. And you feel much more aware of bee lifestyles and biology when you're finished. No matter how much there's going to be something in there that surprises or enlightens you.
Well, we had an episode with Brian Danforth on the publication of the book, and because I was kind of curious how he would take so many disparate pieces of information and synthesize them. All the authors synthesize them into what you describe, it's not a encyclopedia. It is like something that you can read. And he had I can't remember what it was, but he had a writing book that he read. I think it was his book recommendation.
It was How to Write a Lot. I remember. Yes.
That was it.
I listened to that episode, but I have not read that book, which I actually started reading it and felt that whatever it was advising was something I could never accomplish. So I stopped.
Well, it's a great recommendation and a wonderful book. And if people don't have it yet, you really ought to pick it up. It's just don't be intimidated. It is. I was reading Bees of the World recently, just like the introductory chapters, and the print is really small for my old eyes.
Is.
And I will say that Solitary Bees, good font size for.
Yeah, very readable. And then I'm currently reading, I don't know if you can cut this out if you want, but
No.
I'm currently reading The Mind of a Bee by Lars Chitka, which I'm not very far into it yet. I'm still in the beginning sort of sensory processing chapters, but I am reading that one with great anticipation to learn more about just their cognitive abilities and sort of how bees learn and navigate. So
I've never heard of this book.
I'm not far enough along to recommend it yet, but it's what's on on the nightstand.
But so far, so good.
Yeah.
Ah, okay, great. We will put those both in the show notes. And the second question we have is your go-to tool. And I do wonder if it may not be the pan trap anymore.
It is not. I think my go-to tool is the bee squeezer. We use them for so many things. So if anybody hasn't seen or used one, I mean, a queen marking cage may be more familiar, but maybe again, not to everybody. So it's basically a tube or a vial, like anything you could, maybe a 50 ml tube that you could collect a bee in, but it's open on both ends. So it's just a cylinder, and one of those ends is covered with mesh. So you've got an open end that the bee can enter, a mesh end that the bee can be squeezed up to, and then lastly, a sort of foam plunger that you use to press the bee up against that mesh. So it holds it still, it restrains your bee, but it doesn't squish it, it doesn't hurt it. And then once your bee is restrained, you can do, you can take its body temperature, you can paint it with a paint mark, you can glue a radio transmitter on it, any of these things. So that would be my essential tool.
So most of the research that you talked about, the bee, did you call it the bee squeezer?
Yep.
The bee squeezer was central there.
Yes.
Do you make your own bee squeezer, or is it just the queen marking cage?
We have some of each. We have some queen marking cages, and then we have our own that we've made out of like BioQuip aspirator vials that we cut an end off of and put different size meshes over. Sponge pouncers, like for face painting, make pretty good plungers for the foam end. So you can definitely make your own of different shapes and sizes and mesh sizes.
Oh, and listeners, we'd love to see your homemade bee squeezers. Please send those images to me, and I will share them widely. Last question for you is, I imagine it's very difficult, and you work with so many pollinators in so many contexts, but do you have a favorite?
I do. And I'm always a little bit embarrassed that like my favorite bee is one that's super common and drives everybody nuts. But Eastern carpenter bees, Xylocopa virginica.
Oh yeah.
They're big. They're big enough that you feel like you can see their character, their personality, right? Like the males are kind of territorial, they'll come hover in your face. The females will also come hover in your face and look at you it's less clear why they might be doing it. They have fascinating life cycle. Like they overwinter as adults, which is not unheard of, but a little bit weird. They mate a little bit in the fall and then more in the spring.
Oh, also that I didn't know. I thought they did it all in the spring because they're whipping around, but they do a little bit.
They do some in the fall. You'll see mating flights like starting, probably around now. I haven't seen one yet this year, but in like September, October. You don't get all of the big territorial guarding around the nests or around flowers the way you do in the spring, but there's definitely some mating action. They have the males have this very conspicuous sort of yellow or ivory colored spot on their face. Nobody really knows what it's for. In several years ago, Charles Michener wrote a paper on how so many male bees have yellow faces and we don't know what they're for. Carpenter bees, big glowing example of that.
Oh, that's interesting.
They have this weird kind of not exactly social, not exactly solitary behavior where females share nests, but it doesn't seem like it seems like there's really one doing all of the work and then someone else just hanging out waiting for the head bee to have a mishap so she can take over the nest. It doesn't seem like a true like worker queen kind of setup. So they have all of these interesting, quirky things about them that we don't fully understand, even though they're like drilling holes in everybody's back deck.
Well, I imagine an extension as well. It must you must field dozens of calls every year. What do I do?
Mm-hmm. And we don't have fantastic answers. It seems like, if you read existing extension publications, the advice is, spray insecticide on the wood or in the tunnels. Mm-hmm. But for people who want to avoid using insecticides or who are worried about, non-target effects, which I hope would be a lot of people, there's not great advice. And they can ultimately, if you want, I can send you some pictures for the show notes if that's a thing, but they can make a lot of wood disappear. Like I've got x-rays of some benches here on campus showing like the amount of wood that is gone to tunnels. So I don't want to just tell people, oh, don't worry about it. Like it
Yeah.
could eventually do some damage. Yes, it would take many years, but like wood is definitely going away. So we've actually currently got a survey. We're just about to take it down and start analyzing the results, but we've been querying humans around the Southeast about their experiences with carpenter bees. You know, how do you like them? Do you hate them? Why? What methods have you used to try and manage them? Have those methods worked? How expensive have they been? Because it seems like, yeah, there's a ton of questions. The documented solutions are not very satisfying. And so we're trying to get a better like documented picture of just like what is going on out there.
Oh,
So
that's
we
going
can
to
try
be
and
great.
take better first steps toward doing something about it.
Fantastic. And I have to admit, we don't have Eastern carpenter bee. We've got 2 species in the southwest of Oregon, and I'm always getting calls, and I have that same, I didn't quite realize, and maybe our species aren't quite as potentially destructive, but I don't know what to tell people. I read the extension guides, well, paint the wood, and but
and as soon as you say that, like 10 hands in the room go up, like, but they're drilling in my painted XYZ. Like that's clearly
I've heard this too.
Yeah, it's clearly not the solution.
Well, fantastic. A great species. And thank you so much for taking the time to talk with us. You have a very exciting program, and we are looking forward to seeing the findings from your many students and also from your extension program.
All right. Thanks a lot. This has been fun.
Thank you so much for listening. The show is produced by Quinn Sin and Neal, who's a student here at OSU in the New Media Communications program, and the show wouldn't even be possible without the support of the Oregon Legislature, the Foundation for Food and Agricultural Research, and Western SARE. Show notes with links mentioned on each episode are available on the website, which is at pollinationpodcast.oregonstate.edu. I also love hearing from you, and there's several ways to connect with me. The first one is you can visit the website and leave an episode-specific comment. You can suggest a future guest or topic, or ask a question that could be featured in a future episode. But you can do the same things on Twitter, Instagram, or Facebook by visiting the Oregon Bee Project. Thanks so much for listening, and see you next week.